Okay, and then I'm going to start webinar. Cool.
Okay, can I?
Okay. Awesome. All right. Hang on, I don't have my notes pulled up. Okay. Okay.
Don't worry.
Welcome to our, I think this is our 4th or 5th episode of A Conversations. I'm should have had that pulled up. But today we are with Alison Slade from Pfizer, who is going to talk with us today about natural history studies. I think, we're in this time where there's a lot of research and developments happening for not only achondroplasia, but, just other genetic conditions, rare diseases. And we thought it would help the community better understand, how clinical trials and everything start and what the purpose of a natural history study is to sort of help a drug, develop and everything. And so that's what we're here to talk about today with Alison Slade from Pfizer. And so, Alison, thank you for joining us today. To get started, can you first tell us a little bit, what's your role, kind of like what do you do, I guess at Pfizer and your job in general?
Yeah, sure. Thank you. And thanks again, Chandler, for having the opportunity to talk to you about what we do in research and why it's so important, not from a research perspective, but also for the community perspective as well. So yeah, my name's Alison. I'm the global clinical lead for the Reciprocet program, which is specifically designed for achondroplasia, but I'm also a clinical lead for another program within Pfizer's rare disease unit, where we focus only on those diseases and conditions and syndromes which affect a very small minority compared to the general population. And they often have little to no treatment options. So it's a very niche part of what Pfizer does in terms of drug development, but it's something that's very close to my heart from a personal perspective. And I've been working within rare disease for about 5 years. Previous to that, I was in oncology, but the principles are very much the same in that, we're trying to do the best for the community in terms of outcomes for our research, so that when we do develop medicines, they are medicines which are required by the patients themselves. So my background is biology and biochemistry, and primarily what I do within Pfizer is I work on the strategy side in building the actual development plan itself. So we look at literature, what is available from what's been published already about conditions or diseases, and we look for opportunities to develop medicines for that. And that involves really looking at the whole process of how patients get diagnosed in hospitals or in clinics. And then what happens after that diagnosis. If there's no treatment, oftentimes they're either just followed up very rarely within the hospital, or sometimes they have specific procedures and things done. And so we take all of that information and try to build a development plan from first into the human itself, all the way through until the market approval. So my job is primarily focused on the very early part. So I do a lot of the translational research, which means taking the data from what we call preclinical, where it's in animals, and then we translate that into building a human trial. So that's primarily my role within Pfizer, but within the rare disease unit, natural history studies form a huge part of what we do, and we'll talk a bit about why that is.
Great. And I know something exciting for our episode today is Alison actually has some slides that she'll share with us throughout our conversation today. But we do have some questions, to kind of go off of. And I think, we previously discussed, we can kind of like go with them, and then you can show if in your slides, if the answer's what, so we're not too repetitive. So first general question is, and you may have already kind of like just given this, but just for in general, for someone who doesn't know, what is a natural history study?
Yeah, sure. So let me just share my screen quickly, because I think the slides will help to frame the discussion.
And I'll go ahead and I'll put the second question with it, because it kind of it does kind of go with it. But the second one is what is the purpose? So I guess, what is a natural history study and what's the purpose of one?
Yeah. So yeah, I did allude a little bit to that in my initial answer, giving the introduction there. So if you're not aware, there's actually now over 7,000 rare different diseases and conditions and syndromes which exist within literature that have been described. And I mean, many of these indications are where Pfizer can help to develop therapies, but there's very, very little published information about the diseases themselves and what happens to these patients. So a natural history study can help with, as I mentioned, it can help with describing the diagnosis pathway for a patient. So I think with achondroplasia, it's a little bit different now in that it's often diagnosed in utero through ultrasound scans. But for other people who have very rare syndromes, where their symptoms can be very confused with other more common conditions, the diagnosis pathway for them can be extremely hard. And, we often deal with patients and families who wait 5, 6, 7 years for a diagnosis to really understand what's wrong with either themselves or their child. And it's really important for us to understand what happens along that pathway, because part of what we do by collecting that information is we can raise awareness to regulators about how can we diagnose this condition better. So that's one way in some of the information can help. And then the other thing I mentioned was what we do when we collect the study in a natural history format is we really look at all of the different medical investigations that are involved. So that might be in the diagnosis phase itself, but it can also be after that. So, every single thing that you can imagine. So if you have a scan, if you have an X-ray, if you have an ECG of your heart, all of this data comes together either as part of the diagnosis or as part of symptom management. So it's also important for us to understand how many times do people have to go to the hospital to have these investigations? And, that's a huge burden on a lot of patients and their families to have to always go to the hospital and deal with hospitals and have these very sometimes invasive procedures as well. So it's important for us to understand that. And then also any treatments that are given. So that's if any are available. Sometimes and most times there are not anything available for some of these conditions. And then again, it's about us following what happens when they have no treatment. Do they get worse? Do they stabilize? It's about trying to understand how the condition develops through age and through time. If you have quite acute symptoms, for example, which then get better over time, it's important for us to understand how that gets treated. And then, as I mentioned, also it's understanding the quality of life and the burden for the patient and also the caregiver's family as well. And there's two different ways we can collect this information. It can be collected from medical records, what we call a retrospective collection. That generally means that we will have a look at the records and collect the data from there, but we don't follow the patient in any kind of real-time manner. And then the other way that we can do it, if we have patients that are actually coming into the clinic to have specific assessments or therapies done, that's what we call a prospective natural history study. That's actually really the gold standard, because you really have an opportunity to follow a patient from either from the beginning of their diagnosis or through a period of time through their disease course or their condition. And again, that helps us to understand fully the entire journey that a patient has from their diagnosis through their therapy, through symptom management, in some cases also, to when they sadly die. But it's all part of us understanding where can we really add value in terms of a treatment for these patients. So that's kind of the big broad sense of what we do with natural history studies, and a little bit of information about how we use them. But I have some slides on a bit more detail in that a bit later.
Awesome. That's very informative. One of my next one, but I think you kind of, I feel like you kind of already answered all the questions that we're going to ask today, which is great. But what can, and I guess it depends, because I know, I was doing my own research, to prepare for this. Are there different types of natural history studies? Which, I mean, I understand, it can, obviously there are, but I guess like in general, are there, because obviously like a natural history study for achondroplasia would be different than a natural history study maybe for someone who has some rare heart disease or something. But like just in a general sense, I guess, are there different types of natural history studies like in general? If that makes sense.
Exactly. So if I just give you a sense of where we take this data from, that probably help to answer this question. So the first way that we can do this is what we call real world evidence database. And what we do with this is we really interrogate the healthcare space itself in terms of we can look at insurance claims data, so we can see, if I use the US example, how many patients are using ICD codes, which are specific codes which help us to tie all of the symptoms and the diagnosis to one specific patient. So that's one very, it's a very high level overview. There's not a lot of detail there. It's very useful to do things like estimating how many patients there might be in a specific population. It's useful for that. We can have a look at long-term morbidity. So we can have a look at how long patients live with certain conditions. That gives us a very good sense. And we can also look at standards of care as well in some environments. So that gives us an idea, but it doesn't answer all of the questions. So sometimes we start with that, sometimes that comes into the process a little bit later, but that's one type of natural history study that can be done just using medical records. In the rare disease space, it can be very difficult to use these kinds of databases, because when the conditions are very rare, obviously you don't find so many patients, so it can be a little bit harder to identify patients that way, and it doesn't give us all the information we need about what these patients need in terms of a treatment. So it's a good start, but it might be sometimes the only information that you can have, but typically we try to go a little bit deeper when we're looking at a disease, to try to really answer as many questions as possible. So then the next type would be like an academic database. So this could be a database that exists within somewhere like a large academic institute, something like Mount Sinai in California, or something like Nemours Children's Hospital in Delaware. Some of these have very large academic units attached to them, and they themselves hold databases when they become specialist centers for different diseases. So this is data that's collected from patients that are treated normally within the hospital centers that are attached to these academic centers. And it can often be things like a biobank. They can sometimes have patient samples that we can access as well. But again, the data can be very highly variable between institutions. And what that means is then it's very hard to make conclusions when one center says one thing and another center is capturing it slightly different way. Sometimes it's very difficult to tie those two together. And so also it's very limited in terms of the amount of data as well. So it's another type of database that we can use, but we don't use it in its just that format.
Mm-hmm.
The next one that we can do is a patient registry. So this is again a little bit of a layer deeper into specifically how patients are diagnosed and managed within treatment hospitals. So this really involves actually dealing very closely with sites themselves that treat these patients. So you really have to understand where the patients are going for treatment. Sometimes that can just be one center. If, for example, in Europe, you know, in countries where there's just one center for treatment, we would only go to that center because they have all of the data for their patients in that country. In the US, it's a bit more spread out. You tend to have more specialist centers, but that involves, you know, really building a relationship with the site and an investigator there. And it starts to then get a little bit more expensive in terms of funding and resource, because you need more people to be able to get the data out. But that's a good one because we're able to then contact the patients themselves after they've been at the site, and we can ask them questions, you know, if we can use their samples and things like that. So patient registry is another one which is quite helpful. And then I think I mentioned on the first slide that a prospective natural history study is really the gold standard. It's the most comprehensive natural history study we can have. And that really involves following patients over a certain amount of time and collecting that data for a very specific purpose. So it's very highly focused towards the disease or the condition that we're looking at. And it enables us to get really good data from it. But it does require a long-term commitment from patients and from families. And it's also the most expensive way. So those are the sort of 4 ways that we use natural history studies. And as I said, depending on the condition, depends which one of these is the most appropriate.
So, and I think this may have answered my next question, but when I was researching, you know, kind of to get an idea of natural history studies, I came across a blog that was kind of explaining a little bit of the difference, or is there really a difference between a natural history study and what was registry studies? And I guess I was under the impression that like patient registry is different from natural history study maybe, or like, are they 2 different things, or do they kind of like coincide?
Yeah, they do kind of coincide. I think the patient registry part, we tend to use it more within Pfizer once a product is approved. It's more of they really are registries of patients which are available, whereas the natural history study does involve a little bit more, you know, looking for patients where you may not have them before. So they are a little bit different in that respect, but there's a lot of overlap between the 2 as well.
Yeah.
So as I said, they don't tend to, you know, if you're doing a natural history study, you may not need a patient registry because you would overlap too much. But
Right.
it really depends on the condition and the types of registries that exist. Sometimes they exist already, other times they don't. So you know, you really have to look at all of the literature that's available and search on the internet and see if you can find anything that's referenced. Yeah.
Mm-hmm. So what like is so when I guess like who or what decides like we need a natural history study, and like, you know, what's that kind of like, what's the initial step when developing a natural history study? Is it, you know, does it tend to be more of like patient groups seek out to researchers to start that, or kind of like the opposite, you know, like how, I guess if you know, there needed to be a natural history study for a rare disease, like how does that get started?
Yeah, so I mean, typically Pfizer would have already in mind a molecule or a concept for a molecule or a therapy in a specific condition normally. And then what we do is we gather as a team and we start what's called a literature search. And we really go through all of the published databases that are out there, and we start looking for some of the information about, do we find, like for achondroplasia, for example, we would search, what's been published for achondroplasia in the past. And when we started our natural history study for achondroplasia, there actually wasn't very much. There was one paper that had been published, or was just about to be published, but it hadn't been yet. And so we took the decision then that actually it would be a good idea for us to collect our own natural history study and really focus the questions on what we need to answer for this community. And that's very much the same process that we follow for any of the other diseases as well. We sort of look at what's out there, we look at how it's been collected, what kind of information they've collected, and we start to see if there's gaps. And if there's gaps, we might say, well, we need to look at an academic database to fill the gap, or maybe we need to look at some real-world evidence databases to fill the gap. So we start to think about where we might find some other data sources that can. It's a bit like a puzzle, right? You sort of put the picture together, and each step you find another piece, and then you think, oh, okay, now I need to find the next piece. And so it's a bit like just building a puzzle, but it very much starts with, a concept that we have, and we sort of build from there what's available in the published literature. And if we don't have it, then that's where we start to think about where can we find the information that we need. And if we can't find it, then, we need to have a process by which we collect that information.
Cool. And I know you have some more slides to share with us, I think. And just because I want to make sure you have time. So, but one thing I was thinking of, because I know I've been in the past, I've kind of I've had people reach out asking, do you know anyone who could participate in our natural history study? How would you say, because I know sometimes when I guess you do a natural history study, you and when you're enrolling for it, there's different site, I guess, locations. How do you, I guess, decide like what locations you're going to have, like where participants can enroll? Obviously in the perfect world, I guess we would have natural history studies at all hospitals or children hospitals or whatever, across the US or across the world, but that's not the case. So what, goes into, I guess, deciding locations? Is it more like would Pfizer reach out to locations, or do you kind of, let researchers know that you're doing this and then give them the opportunity to say, yes, we would like to be a host site for natural history study? How does that kind of happen?
Yeah, it's a little bit of both. So typically what we do again, once we have a concept in place and we start looking into the literature, we, as part of that process, start to identify what we call key opinion leaders for specific conditions. So there's always somebody whose life's work has been dedicated to, these diseases and conditions. So what we do is we find those people first, and then we sort of talk to them, and we just say, we have this idea as a concept for a treatment. We'd like to, understand a bit more about the community, about the disease, the course, the process, the diagnosis pathway, everything. And that generally starts a dialogue of, where they find patients from. How do they get patients funneled to their sites? As I mentioned, in Europe, sometimes in smaller countries like Italy or very small countries like the Netherlands and Belgium, Austria, there'll just be a single site actually where all of the patients, if they get diagnosed with a condition, they just go to that one site. So when you get to that site, you hit the jackpot. In the US, it's obviously a little bit different because you need to be more geographically spread. The country's obviously much bigger. So And then, once we've engaged with our key opinion leaders, we often will ask them for, recommendations of other, opinion leaders or doctors that they work with who, would be able to provide a lot of data, or patients, or who would be very specifically interested in trying to join. So we would reach out to those people, and then sometimes we get approached also by other centres where they've heard that we're doing some research, and they reach out and said, well, we'd like to participate still. So it's a little bit of both, but it generally starts with Pfizer itself identifying these what we call key opinion leaders for different conditions.
Cool. Well, I feel like you've answered all questions that I was going to ask. So if you want to go through your very informative slides that we have for our audience, you're welcome to do so.
Yeah, I mean, it's just giving a little bit more information about why we choose different designs for different conditions. So I think I already mentioned some of this anyway, that really the design that we pick ultimately, whether it's a prospective one where the patients are coming in over time, or whether it's a retrospective one where we're looking back in the medical records, it's really it's a decision which depends on the feasibility of that, in terms of is there enough patients around to be able to look at that, and can we collect that information? It's also availability of resources on Pfizer's side, because it does require a different amount of resource to run these studies because they're slightly different design. And also the time it takes. Prospective studies much harder to set up, it takes longer. A retrospective study, because you're just looking in the records, you don't need to speak to patients necessarily. They can be much faster to complete. So kind of all of those things we sort of weigh up. It's a balance of, we want the information, but we also need it in a timely manner. So those are sort of the three pillars in which we sort of have a look at about which of the designs is most appropriate. And then, as I said, a prospective one is enrolling patients over a period of time. It's very comprehensive data collection. We can use the patients identified for treatment trials in the future as well. So it's very helpful to identify patients in that respect. It does take longer to recruit, and it's more costly and resource intensive. But the retrospective studies, you're using existing data that is already available. It's much faster, it's more efficient, but it can be challenging to sometimes find the exact data that you need. And that often sometimes means you have to go to lots of different places instead of just one place. So yeah, so lots of advantages and disadvantages. As I said, depending on the condition, depends on how we decide which way we want to go. And then just to give you an idea about how we apply the information that we collect from the natural history studies. So it really helps us facilitate the overall drug development process, because oftentimes when we submit to regulatory authorities, a lot of the time they can be very naive about the condition or the disease itself. So Pfizer often has to go through a process of educating the regulator about what is this condition and how burdensome it is for patients, and there is no treatment. So there's also a level of us having to educate people like the FDA about conditions, and the natural history studies can be very good as evidence to supply to people like the FDA, for example. And it can also help reduce the amount of regulatory hurdles as well. So if they understand the severity of the condition and how burdensome it is for the population, they may be more open to faster trial designs or more innovative therapies in order to get treatment out to the community faster. So that's one big way that it gets used. When we're designing the trials itself, as I mentioned, we can sometimes be a bit more innovative and a bit faster in the design. We can also sometimes reduce the need for a placebo, which of course is great for patients who are desperately looking for treatment. So that's one way that it can be used. We use it to define the target population in the condition or the disease itself. So lots of different genetic diseases, they also have very small genetic subtypes within the condition, and those subtypes may respond differently to treatment. It's very hard to know. So it's important for us to make sure that when we're developing a therapy, we really understand which of the patient populations will benefit the best from it. And then, you know, how can we find the patients? That's sort of alluding to what you mentioned about, you know, where do the participants come from? You know, part of our natural history study is also, you know, how do these patients get diagnosed? Can we recruit them from other places within the country? Trying to understand that aspect of it. Developing biomarkers. This is actually probably one of the most important, in that when you don't understand a disease fully, it's important that you can understand if there's any biomarkers in the blood or anything that you can watch change over time. And with these rare conditions and diseases, a lot of the time nobody knows what those are. So if I take achondroplasia, for example, we often use collagen X. Collagen 10 is a fragment of collagen which is produced when bone.
You froze up on me. Oh,
Sorry, I'm back again.
oh,
Sorry, I lost you.
You're good. You're good.
Can you hear me? Okay.
Yeah.
So where was I? Yeah, biomarkers. So that's one. And then understanding how we can collect them from patients as well. So, you know, in children, for example, lots of children don't like having blood samples taken. So, you know, one of the things might be that we collect just finger prick samples instead of blood from the arm, from the veins, because that's much more friendly for the patients. So it's good for us to understand things like that. It's much easier to collect. Developing clinical outcome measures. So what this really means is we really want to identify with each condition or disease what is really most bothersome for those patients that they need to have treated. So yeah, I mean, it could be patients who have epilepsy, for example, a rare type of epilepsy. It might be to really reduce the number of seizures that they have, to recover from seizures faster. So we really have to understand, you know, what is it that the patient community actually want in terms of a treatment. And, you know, if you take epilepsy as an example, you know, obviously nobody wants to have seizures, right? So that's going to be one of the key outcomes that you want from your trial. And then, as I mentioned, that, you know, we can also use them as historical controls. What that means is, when we get to a trial, we have to compare an untreated population with a treated population. And natural history studies can sometimes serve as that untreated population that we can compare to. So again, that can also mean that we don't have to have as many placebos required in a treatment setting as well. So those are all just given little examples of how we use natural history studies and why they're so important for us as an organization. I think that was my last slide.
We did get one, well, it's not one question.
Oh, yeah.
Some person, we did have someone kind of submit a few detailed questions, and I'm not sure if we're going to be able to answer it too well, just because our main focus of today's conversation is really just about natural history studies in general, but just maybe if you can provide any insight to it. What about the children who are currently measured in Theracon trials, which we know is Rouseff-Roussette, and by the end of the year, I guess if they're currently, I'm under the impression that they're currently in the measurement study, maybe for a Rouseff-Roussette trial, but by the end of the year are looking to start taking BioMarin's product, Voxzogo. And then what about your trials Pfizer's trials, when probably all of the patients, I guess, when will they leave the program, I guess, graduate from the program? And then I would like to know if the research will continue as fast as it is, or if it can be affected from this event. In Italy, we have multiple hospital center for achondroplasia, but only the hospital in Rome has been selected as a site where children are tested with this medicine. Having more sites involved could help Pfizer to have, I guess, patients from leaving to go start taking Voxzogo, which has already been approved.
Yeah, exactly. Lots of questions within that question. So I'll start with the first part. I have it on the screen here in front of me. So
Cool, great.
Yeah, so I mean, the children that are involved in the current natural history study for Rizipacect, as you know, we're collecting that data as it. You have to see it as its own protocol. So the data that we're collecting from that is for understanding the natural history of achondroplasia. So and I know that there are other natural history studies as well collecting similar information, probably slightly different from us as well. So if you are moving into a treatment study like Biomarkers, then you would then just finish your participation in the natural history study of our one, the 𝘾𝙊𝙉𝙍𝙑𝙀𝙁, if you're going into a treatment trial. So that's the way the protocol is currently designed. I don't think we will change that right now, but we have these ongoing discussions all the time. So but as it stands right now, if you move on to a treatment study, you would finish your participation in the 𝘾𝙊𝙉𝙍𝙑. And then if I answer the last question, so the 𝘾𝙊𝙉𝙍 study itself, we follow the children for 5 years, unless they leave earlier to go on to a treatment. So that's also fine. But we follow them for a maximum of 5 years. And then what's the next question here? What about your trials when probably all of the patients will leave the program? Yeah, I mean, I think what's important for you as families have to discuss with each of your own treating physicians and investigators about what is the best option for your child as it is right now. So it's hard for Pfizer to predict what will happen in the future. We have to look at the data, and the data may be good and it may be bad. We don't know yet. So I think I would just encourage you, if you're not sure, is to have ongoing discussions with with your own doctors to discuss what's the best for your child. And then what was the next one? Yeah, so in Italy, it was a poor example for achondroplasia because we do have more than one site that has a lot of children in the natural history study. I think for the phase 2 study, I think what was very important for us is there's a huge variability in the way that people measure children. And so in order to give the program the very best possibility of getting good data, it's better to have a smaller number of sites initially, because you can very much easier control for variability in the way people measure. So if you have the same people doing the same types of measurements over and over again, they tend to get better at it and they understand the whole process. When you have lots of people doing measurements and not so many patients, because there's more sites, so there's less patients available for each site, you start to see a lot more variability in measurements. And then, of course, you run the risk that you show that your product is not working, but actually it's just because you've got lots of variability in the measurements, which is not helpful. So in the phase 2, we did take a decision to have a lot less sites than what we needed, purely to control for, or to try to control for that variability, to really use sites that were very experienced with doing the measurements, In the phase 3, obviously we'll need more patients, so there will be more opportunity for sites to participate then. I don't know which sites will be selected right now, but there will be more sites available for the phase 3 program. But yeah, for the phase 2, we kept it much smaller. Yeah. I think, was that all of the questions?
I think so. That was I feel like that was a really good way to kind of conclude this. And I want to, and to kind of go off of what you had said before answering the last question, it's for any families who watch this later, it is, I think it's always a good reminder that families know, each family's different, and you have to go with what is best for, you feel like, your child, and kind of in your family dynamic. We all have different lifestyles, day-to-days, and so I think it's just a good reminder that it's good with all of these new research and different trials that we have happening within the, I would say, achondroplasia world community. You may start one thing and then realize being part of another is better for your whole dynamic. And I think it's important for families to know that's okay. It's okay if you change or decide you no longer want to do something or whatever. At the end of the day, you're making the best decision for your child and your family. And so we know it's tough, obviously, but yeah. So I think that concludes our conversation. It was thank you, Alison, so much. It was I personally, I feel like I've always kind of had a general idea of natural history studies, just kind of with being so involved over the past few years, but I've actually never had kind of a deep dive conversation about it, or had someone really fully explain it to me in detail. So as a patient advocate myself, I feel like I very much benefited from this conversation today, and hopefully parents, anyone who watches this recording later, kind of understands more why we have to have natural history studies in order to, create and develop future possible treatments for achondroplasia or any conditions and rare diseases. So I think this was a really, I benefited from this conversation a lot. So hopefully our audience does as well. And so thank you so much.
No problem at all. So if you have other questions that come in later, then you know where to find me. And,
I think this would be,
and you have the slides, so you can also share those as needed as well. So.
Definitely. And I think this would definitely be a great conversation to maybe have later this year, if there's more kind of involvement in natural history studies. We'll have to see, but we would love to have another conversation with you, and we will keep everyone up to date and posted, and we will have this conversation on our Growing Stronger YouTube and Facebook page as well, just for anyone watching who wants to refer back to this later. But again, Alison, thank you so much. And
thank you all, and yeah, wish you a great weekend.
Thanks, you too.
Preparing for
happy Friday, everyone.
Yeah, happy Friday, and yeah, have a great weekend, and I'll see you soon, hopefully.
Awesome. Thank you.
Thank you. Bye.